Hi, everyone. This is Richard Jeffries from Treatment Action Group, and I'm here with Nelson Vijay from Program for Wellness Restoration. Thanks so much for joining the webinar today on suboptimal immune recovery on antiretroviral therapy. Just a few logistical things to start. As you probably heard, we're going to try and record today's webinar. So to begin with, the audio for everybody will be muted just to minimize the background noise. There is the option to ask questions as we go along via the chat function, and I'll try and keep an eye on that and respond to questions or respond to them at the end of the presentation. And also, hopefully, at the end of the presentation, we can open the audio up for anyone that wants to ask questions via audio. And just to sort of give a little bit of an introduction, obviously, thankfully over the past couple of decades, we've seen antiretroviral therapy improve and it works incredibly well. And thankfully, the newer treatments have become more tolerable. But there is still an issue where some people, particularly individuals who start antiretroviral therapy later, are at risk for not getting really great sort of recovery of their immune system. And so the sort of outline of what we're going to talk about a little bit today is just what kind of happens with the immune system when HIV is suppressed by antiretroviral therapy, what kind of factors have been reported that are associated with actually having a persistently low CD4 count and sort of poor immune recovery despite the antiretroviral therapy effectively suppressing HIV viral load. We'll describe a few studies that have found that there is, although the absolute risk of illness, I want to be very clear, declines very dramatically thanks to antiretroviral therapy, and that sort of applies to everybody. The relative risk of morbidity and mortality, which is the kind of medical term for illness and death, is slightly increased in people that have lower CD4 counts and that don't get good CD4 recovery on antiretroviral therapy. And then next we'll review some of the candidate therapies that are being looked at or are being looked at to see if they can improve the recovery of the immune system when they're added to antiretroviral therapy. And then lastly, Nelson will describe a little bit about a meeting he initiated with the U.S. Food and Drug Administration, who sort of regulates the development of approval of candidate therapies, about potential ways that there might be to try and encourage development of treatments for this particular population. And so just to move into the initial presentation parts, when antiretroviral therapy, the sort of triple combinations that we're familiar with now, first came along in the mid-1990s, there were studies kind of pretty quickly looking at what happened to people's immune systems when they began on these combinations and the HIV viral load was suppressed. And what these studies found is that there's usually a pretty quick increase in the numbers of a particular type of CD4 T cell called a memory T cell that sort of reappears in the blood in people that had seen them decline due to their untreated infection. And the studies found that this is partly because some of those cells had been trapped in the lymphoid tissue sort of and weren't circulating in the blood. And also when HIV viral load is suppressed, these cells were able to proliferate and sort of restore some of their numbers. And this is important because the job of memory CD4 T cells is to respond to or control infections that you've been exposed to in the past. So they're sort of very important for preventing the pathogens that can cause problems like thrush or pneumocystis carinii pneumonia, which I think is called Girovetti pneumonia now. But all those kinds of pathogens are controlled by memory T cells. So it's important that those were sort of restored quickly. The other thing that was seen in these early studies is they can measure the immune response to these kinds of opportunistic pathogens, and they were found to improve. And also people were able to respond again to sort of routine vaccinations that people get for flu and so on. There's also, they saw a very quite dramatically decreased sort of overactivation of the immune system that was being caused by the replication that the antiretroviral therapy was able to reduce or stop. And then lastly, the sort of slower part of the immune reconstitution that was seen in these early studies is a population of CD4 T cells that are called naive T cells, which, as the name suggests, they're the kinds of cells you need to respond to new pathogens or new vaccinations that you receive. And they're produced, sort of the production of T cells starts in the bone marrow, and then they get educated in this organ called the thymus, which is located just behind the breastbone. That's the kind of training camp for T cells, and that sort of produces new naive T cells. But it does so most rapidly in childhood, and it slows down a lot as we get older, and so you sort of lose your naivety, which you kind of do anyway as you get older, but you lose it immunologically as well because of the slow production of these cells. So it took them, when they were studying people experiencing immune recovery because of antiretroviral therapy, these cells came back, but quite slowly and over a long period. And so this next slide, I think it's from an AIDS clinical trials group study in the US, and I just wanted to show it just to give a sense of this recovery in CD4 T cells that's seen. It quickly became evident that it was quite dependent on where someone was when they started antiretroviral therapy. So the lowest dotted line on this graph is people who started with very low CD4 T cell counts of between 0 and 50. And as you can see over this period, 144 weeks or close to 3 years, they were seeing increases for the most part, but quite slow. And it's really the people towards the top who started with more than 500 or between 350 and 500 who experienced the better gains, and the lines represent the different sort of groups based on where they started. The gray shading is where the sort of range of normal CD4 T cell counts for comparable HIV negative individuals is, and you can kind of see that over this 3-year period, it was only the people that started with the higher CD4 counts that were kind of getting back into that normal range. Although longer-term studies have found that that this can continue, and more people do start to get into that normal range. And then the problem that we're sort of talking about today is that studies then sort of began to see that there are a subset of individuals who really experience CD4 T cell recovery that's not optimal. Sometimes it doesn't seem to improve at all, or it improves very little and very slowly. Unfortunately, there's not really a clear consensus as to how exactly you define what constitutes suboptimal in this context. There's been quite a lot of slightly different definitions used in the studies that are conducted, but the general sort of theme is that it's people who experience, don't experience an increase to above 200 CD4 cells, or some studies have used 350, but it's generally just this persistent low CD4 count that doesn't get back to what the normal range would be for an HIV negative individual. And when you sort of look at the studies that have been published overall, the estimates are sort of in the range of maybe 15 to 30% of individuals who start on an antiretroviral therapy regimen and are adherent and get good suppression of viral load will experience this phenomenon of not getting great CD4 T cell recovery. And just to look at a couple of specific studies that have tried to sort of quantify the phenomenon in a large number of individuals, there's this analysis of 2 large international cohorts called the ART Collaboration and Cohere. They specifically looked at individuals in their cohorts who began ART with less than 200 CD4 T cells, and out of the 5,550 individuals they looked at, there were 835, or 15%, who after 3 years of HIV suppression still hadn't managed to see an increase of CD4 T cells to over 200. And there's quite a lot of published studies on this, and it's been seen in countries from all around the world. Just to provide another example from a cohort in low-income countries, there was a report that it was about 17% of people, the follow-up was kind of shorter term, but they just didn't experience any CD4 gain even after 6 months of HIV suppression. And so it is the sort of a phenomenon that's seen everywhere. And then just to sort of look a little bit longer term, this is another ATTG study. There's some estimation that's gone on in here, but I think the sort of important parts are down towards the bottom of the table, which is just that really the biggest risk factor for not getting a robust CD4 increase over this 7-year period was starting at lower levels. So people that began with less than 200, 47% was still below 500 at 7 years, and it was pretty similar for people that were started between 201 and 350. And there's been quite a lot of work to look at what kind of factors might be contributing to this, and to help understand sort of what the mechanism is that might be explain this. The most consistent things you see in the studies are older age, and as I've gone over, the CD4 T cell count when somebody starts antiretroviral therapy. And I think the thought is that the link with older age is because of this phenomenon of our production of new T cells slowing down as we get older. It sort of increases the risk that an older person beginning antiretroviral therapy is not going to have so much reserve of new T cell production to help contribute to immune recovery. Again, there's a lot of individual variability, so some people do get very quick recovery even if they start low, but there is this subset of individuals that have this persistent issue. And the low T cell production is one thing that's been seen. There's also a kind of overstimulation of the immune system that can continue despite HIV suppression. And some studies have linked that to co-infections such as hepatitis C, although there's some contradictory reports on that. And also this phenomenon of leaking of what would normally be friendly bacteria that we have in our gut into the circulation. That's called microbial translocation, and that does seem to be seen more frequently and persistently in people that experience suboptimal T cell recovery. And so some of the therapeutic approaches have been looking at that. There's also a potential for scarring damage to the lymph tissue as a consequence of, because that's where HIV primarily replicates. It can cause damage to the tissues, and the role of that tissue is to provide sustenance to T cells. The tissue cells produce proteins that help with CD4 T cell survival, and if there's a lot of this fibrosis, scarring damage, the sustenance provided to CD4 T cells is inadequate, and it can contribute to a greater frequency of CD4 T cell death or apoptosis, which has also been documented in people with suboptimal CD4 T cell recovery. There are some genetic factors that have been reported, and very recently in a study published just this year, it seems possible that there's some people that actually make antibodies that may cause the premature death of their own CD4 T cells, and that's a relatively new finding, so that's just starting to be looked at now. And then shifting to what's known about how this poor immune recovery might link to health, and the sort of worrying aspects about this, and why there's concern about the need to potentially develop adjunctive treatments to help address it, is that it does seem to be associated with a somewhat increased risk of illness and death in the studies that have looked at those endpoints. But I think, as I said at the beginning, because We don't want to sort of make people think that there's a need to panic about having low CD4 cells necessarily. That the absolute risk of something bad happening, even if someone's CD4 T cells are quite low with HIV suppression, is quite small. It's just that relatively, compared to someone with a higher CD4 gain, it's somewhat elevated. And everybody that gets HIV suppression does see their risk of illness and death, dramatically decrease, which I think is important to stress. But the relative risk does seem to be elevated around 2 to 3-fold in somebody with suboptimal recovery versus somebody with greater CD4 gains. And just to provide a specific example, in the large cohort study I mentioned previously, the number of people that died, there was about 7.9% of the people that had persistently below 200 CD4 T cells versus 2.3% of the people who recovered to higher levels. So that was a significant difference, even though compared to the size of the cohort, it wasn't a huge number of people. And then this is just a sort of another study at the Dutch Athena cohort, looking at people with different CD4 counts on antiretroviral therapy and their risk of a variety of different endpoints, including cardiovascular disease, AIDS events, non-AIDS events. And the red line is the people that were persistently below 200 cells, and they had the fact that the top line is showing that they had a greater risk of these things happening. The sidebar is the percentage of people that had an issue. I would stress, though, that the data really was, I think, most collected up until 5 years, and so I think it's roughly 2-3-fold increase in risk compared to the people with higher CD4s. That huge and slightly scary spike towards the end, I think, is based on estimates and a few numbers. So I don't think I would not panic about that, and we would look more just at the five-year mark. And so all of that sort of background is the reason why there has been interest in trying to develop therapies that might help promote immune recovery on the basis that would further reduce any excess risk of morbidity and mortality. And really one of the first that was developed at the National Institutes of Health and studied in 2 very large, thousands of people, efficacy trials, is interleukin-2. It's an immune system messenger protein that promotes the proliferation of CD4 T cells and was shown to increase CD4 T cell counts in people with HIV on antiretroviral therapy quite dramatically. But when they looked at whether that increase was associated with a health benefit, unfortunately, the evidence was that it really didn't have a significant effect compared to just antiretroviral therapy. They also looked at whether the CD4 cells, as a way to look at whether the CD4 cells that were being created were functional. They looked at how people responded to routine vaccinations, and it didn't seem to affect that response. And eventually there was evidence that the CD4 T cell subsets that were increasing were maybe not the most beneficial. Some of them were what they call regulatory T cells that are more involved in kind of suppressing immune responses. And the bolded part at the bottom is just what I think the big lesson has been from these IL-2 studies, which is that not all CD4 T cells are necessarily created equal, and that really to be sure that there's a health benefit from an immune-based therapy as an addition to antiretroviral therapy, it really has to be proven that it has a positive effect on the health, and you can't just assume that there's going to be a benefit based on a CD4 T cell count increase. And that's kind of makes it challenging to try and develop new therapies. So another cytokine therapy that's been quite extensively studied, which works a little differently from IL-2, is interleukin-7 or IL-7. There's evidence that perhaps it promotes CD4 T cell increases in a slightly better way, and then more the kind of right sort of CD4 T cells, although that is not proven. And there was some evidence from studies that it also reduced levels of kind of overactivation of the immune system and this inflammation that can persist despite antiretroviral therapy. The possible concern that it was also maybe increasing HIV in some CD4 T cells by causing them to proliferate, although those increases were very slight. And the original manufacturer was a French company called Therapies, and they did have quite ambitious plans to conduct studies in people with suboptimal immune recovery, which would have really proven whether or not it could benefit health. But before they were able to start those studies, they went bankrupt, unfortunately, in 2015. So it's a little unclear what's going on now. The rights are said to be with the French ANRS, but whether there's going to be additional studies is unclear. I was informed just the other day by Irini Soretti at the National Institutes of Health that there's also a long-acting formulation of interleukin-7 being developed by a Korean company called Neo Immune Tech, and it's being studied in people with other kinds of immune deficiencies, but there hasn't been interest in studying in HIV as yet, but that may be something that there's potential to encourage. A gene therapy which has shown some efficacy, you may have read about it. It's a therapy developed by a company called Sangamo Biosciences. It's quite a complicated therapy to administer. It involves extracting CD4 T cells from somebody via a process called apheresis, where people get hooked up to a machine to get their CD4 T cells extracted. The CD4 T cells are modified in the laboratory to remove the CCR5 receptor that HIV uses as a sort of latch to gain entry into CD4 T cells. And the cells are then expanded and reinfused into that same individual. And one of the first trials was in people with suboptimal immune recovery, and it showed quite dramatic. It was just a single administration, but it showed quite dramatic increases in CD4 T cells, at least for a year or so of follow-up. And there were improvements in the CD4 CD8 T cell ratio as well, which is another kind of marker that's used to assess immune function. There was quite a big advocacy effort to try and encourage the company to continue to develop the therapy for this population, but unfortunately they haven't really shown interest in doing that. There was a letter from quite a lot of activists that was sent, but I don't think they ever even respond to. And there was one meeting which unfortunately wasn't very successful in terms of getting them to change that position. So although the therapy is still out there, it doesn't seem to be being looked at anymore in this population. And then there's a variety of other candidates that haven't really had they're not supported by any particular company, so they're being looked at by academic university researchers that have played a really big role actually in continuing to look at this issue and explore potential interventions that might be helpful. There's been quite a few studies of probiotics that are intended to promote gut health, and there's some reports that they have kind of effects in reducing inflammation markers and possibly other benefits related to gut health. They haven't really seen particularly significant CD4 count increases, and they haven't really been looked at. There's been a few studies, but not too many in people with suboptimal immune recovery. There is an ongoing study of a probiotic called BCL3 that's happening in Canada, which is a quite carefully designed trial that's hoping to really assess whether there are benefits from probiotics, which I think is kind of crucial, because although they're available to people, they're often kind of hideously expensive, and so and there are some concerns about potential risks, although they haven't really been seen in very much in studies, and there needs to be some sort of evidence if it's going to be recommended that people try that approach as a means to promote immune reconstitution. Mesenchymal, I'm not even sure how to pronounce that, stem cells were looked at by a group of researchers in China. they're quite difficult, I think, to sort of cultivate. They can come from a variety of sources, including fat tissue and umbilical cords. And so they may not be the most practical approach, but the small pilot study they conducted did report CD4 gains in people with suboptimal immune recovery on ART and some anti-inflammatory effects. And there is a larger trial that's now being conducted, and also a study ongoing in Spain to look at that particular approach. Pyridostigmine, I think that's how it's pronounced, is an approved treatment. Again, it's another small pilot study that was conducted that suggested some benefit, and there's a larger trial ongoing now. It's an FDA-approved drug for myasthenia gravis. The issue of fibrosis, the scarring damage to the lymph tissue, has led to the development of antifibrotic drugs as a potential intervention, and they have shown some potential in a published study that involved macaques treated with infected with simian immunodeficiency virus and treated with antiretroviral therapy. And there's now 2 ongoing trials in people on ART with CD4 cells below 600. Lastly, I don't know really anything about this particular nutritional supplement, but there is a small study happening that's targeted to people with persistently low CD4 T cells at the University of Southern California. And those are sort of the kinds of things that are happening right now. There have been other approaches tested. The AIDS clinical trial group in the US looked at something called keratinocyte growth factor, which was suggested to maybe improve the function of the thymus and production of naive cells, but it didn't really show significant effects. And there are some other things that have been looked at that didn't really seem to have the desired effects. But the research thankfully has not quite ground to a halt. There are things still being looked at. And I'm now going to hand off to Nelson Vogel to talk a little bit about a meeting that he initiated. It was kind of over a year ago now, but it was just to try and start to look at perhaps some possibilities of encouraging the development of therapies for people with suboptimal immune recovery.
Well, thank you so much, Richard, and thanks to everybody. We have close to over 50 people right now on the call, so we're really happy with this turnout. A little bit about myself, although I think I'm looking at names here, and a lot of people know who I am. But I've been HIV positive for probably close to 34 years. I have multi-drug resistance to pretty much every class of drugs, including integrase. And I was just lucky to join a study 6 years ago, using ibalizumab, which is a drug that may get approved soon within the next 3-4 months. And we did some advocacy work to actually help the company apply for an orphan drug indication. So I'm going to explain what that means and what the incentives are for industry to proceed with work on the immune reconstitution area. As you heard from Richard, one went out of business, a small company, and another one basically walked away from this indication. So we've already seen some promising data from some options, and yet nothing has come out of it. And I think the main issue here is that companies are afraid to be required to do large studies to prove that the new CD4 cells actually increase survival. And as we know, in 2017, it will be basically impossible to do a large study that measures survival as an endpoint. So Richard's actually going to talk about what other endpoints we can look at, so that we don't have to do a 5,000 people study for over 5 years like interleukin-2. So back in last year, a few of us, I think, and I think Matt Sharp is on the call. I saw his name. We were very encouraged over 2 years ago about the results from Sangamo, from their therapy. Matt is actually an activist, also part of the ATAC, AIDS Treatment Activist Coalition Group. Back then he was. And he had really a great response on CD4s. His CD4s were, I think under 300 when he started, and they went up all the way to 700, 800. I'm misquoting Matt, but I was very jealous because my CD4s are under 300, and I have been so for a while, for a long time. So that encouraged us to advocate to go to talk to the company and see if they were interested in developing something for people with lower CD4s. And obviously, Richard told you guys about the difficulties we had to get that through. We're not giving up on that, by the way. The reason we're doing this call is to get interest and motivate people to get involved in this activist work. I think, as you all know, I'm a patient with multi-drug resistance. There's a lot of good things have happened for patients with what we used to call salvage patients with multi-drug resistance in the last 5 years. And I really think that this population that we're talking about today is a forgotten minority, although I don't think at 20%, and I will be talking about that 20% size of the population is a minority, but really is a population that nobody's really developing anything for. So I think we, as a community, we can change that. So we decided, I reached out to the FDA to see if they were interested in discussing with activists. Basically, there was no industry people there. About what we can do to remove barriers, especially for small companies, small biotech companies that may not have the funds of large companies to do studies. And they were very, very open and friendly. I think we have very good friends actually at the FDA. They have been helping the community for over 25 years. So the notes of the meeting are available on the treatmentactiongroup.org website. And by the way, that's Richard's nonprofit. He works for that nonprofit. I encourage everybody to go on that website because there's a lot of good info there. So I wrote down, I actually tried to assess how many people are in the United States like me, or like Matt Sharp was too, with CD4s under 300, or in this case, it's hard to say because some data shows that under 200 is what we have as data, but I do say that there's some data also that shows increased risk of morbidity and mortality for people with under 350. So the next one, we're going to go through the first part of Richard. I need to move this. Can you click, Rich? Okay, there you go. So, so I was trying to convince the FDA that there are under 200,000 of us, and why is that? Because an orphan drug indication, and I'm going to explain what that means, really applies to a population that is under 200,000 in the United States, and we're talking about U.S. only. So that was my goal because I really thought they were going to be under 200,000. And what's the advantage of having an orphan drug indication? Well, there are many advantages. One is tax credits, which, by the way, that may be removed on the new tax bill. I just read that a few days ago, so that's a scary thought that that bill may remove the tax credits for orphan drug indication. The number of patients required to get an approval is much lower for orphan drug indication, and also is the requirements for funding. So I thought it would be a perfect angle to approach especially for smaller companies like Sangamo and all the other ones that do not have the funding, right? We have good news. We were able to work with the FDA in the past 2 years to actually prove also that people with multi-drug resistance in this era, also there's less than 200,000 of us. So ibalizumab, and I'm going to show you that later. Ibalizumab is a drug I'm using. It's an IV every 2 weeks, and that actually will be approved as an orphan drug. The first antiretroviral, if you want to call it, it's really a monoclonal antibody. It will be approved in the next few months as the first orphan drug indication for HIV. So a goal that we have is to also obviously have the same kind of indication for what we call the INR, the immunological non-responders. I'm sorry that we use a kind of we always use in terms like MDR as multi-drug resistance and INR as immunological non-responders. But anyways, let's not digress. How do we come up with a number? Well, this is data from 2014 from the CDC website, and it's kind of scary numbers. They're probably around 1.3 to 1.4 million of us in the United States. 86% diagnosed, so a bunch of people don't know they have HIV. They're 40% engaged in care. I think that number may be improving. Obviously, this is 2014 data. 37% of the ones are engaged in, the total population is prescribed antiretrovirals. And the saddest part of this data is that 30% of those patients in the United States had viral suppression. And hopefully in 2017, we'll see a better number there. So let's go to the next one. So I'm sorry, before we go. So 30% of 1.3 million is 390,000 people. Those are the ones that are virally suppressed. And we really are talking about focusing on people that have been virally suppressed for at least 3 to 5 years. So they're taking their meds, they're going to the doctor, but their doctor is telling them, listen, your CD4s are, where they are, and there's not much we can do about it. That's the way it is. So those are the people we're focusing on. Around 20% of the 390,000, and Richard showed a little bit of that data, may have suboptimal immune recovery. So that's 78,000 people. So that's good because that's under 200,000. So that actually justifies advocating for orphan drug. And the FDA completely agree with us on that. So that was not a hard sell. So the most recent CDC Surveillance Report states that at least in 2015, 21.6% of the new infections were classified as stage 3 AIDS, meaning they had lower than 200 T cells, so around almost a quarter of people coming in for diagnosis, either at the emergency room or because they're getting sick or something, already have AIDS. And it has decreased a little bit from 2013. It used to be 23.6, and in 2015, 21.6. So who knows? Maybe in 2017 will be at 20%, and hopefully that will improve with time. I really hope so. A conservative estimate would be that 20% of these late diagnosed individuals may become INRs. So that would be anywhere from 1,500 to 2,000 new people coming every year diagnosed with low CD4 counts. Okay, so that is a population a new population that keeps adding on to those of us that have been living with HIV for a while and have under 300 CD4s. And this trend should hopefully be able to decline in the future, depending on how well we do our work with outreach and education. But at the current rate, it is extremely unlikely that there will be over 200,000 people, HIV positive people, with suboptimal immune recovery. So once again, the orphan drug designation, it's more than well justified for this field. And orphan drug is not a new thing in HIV. I was actually surprised when I did this research last year that there were a few companies from even from the 90s, like 1990, one of them in that table. I'm not going to go through all this, obviously, that got designations. And by the way, saying that something has an orphan drug designation is very different than saying something got orphan drug approval, because designation is something that is given maybe in phase 3 or phase 2 or even phase 1, sometimes, or mostly phase 2. But a company can withdraw the application. FDA could approve the application and the approval of the NDA, or they could just walk away from it. But they're having a few products, not HIV treatments per se, but mostly for comorbidities or mortality-related issues that have been designated as orphan. As I said, the next, let me see the next slide. Yeah, you even have things like testosterone and thalidomide. The companies actually applied for the designation, but none of these companies, and even tramadol, which is a painkiller for neuropathy. But we haven't had yet an approved drug that actually treats HIV, and as I said, that will be coming up soon, since the company TaiMed got orphan drug indication for ibalizumab, which is the drug that basically I would say saved my life, but my CD4s have not increased even though I'm virally suppressed. So I do have a lot of motivation for this project to go ahead for obvious reasons. So this is where I'm going to have Richard take over. He's really and as you can tell, everybody that doesn't know Richard, he's really our in-house immune recovery and a biologic treatment expert in the activist world. So we're very happy to have his focus on this work. So Richard, take over from here. Thank you.
Thanks, Nelson, and thanks for that presentation. That's great. Just to sort of touch on one of the other things that was discussed or this FDA meeting was sort of whether there might be novel ways to try and assess treatments for this population that would require smaller, not quite so huge sample sizes. And I think, there's a bunch of biological markers that you'll see that get looked at in quite a lot of different studies that would be kind of the sort of fairly obvious candidates that I've just listed here. There's particular interest among some of the researchers in things that don't just measure numbers, but try and give some sense of how well the immune system is working. And really one of the only practical ways of doing that is this idea of using, immunizations and seeing what kind of response people mount to that. So that might be a sort of a relatively novel candidate for inclusion in these types of studies, along with other markers of immune system function and inflammation and those kinds of things. The other potential options, there's a fairly new sort of idea, and the FDA's been doing quite a bit of work, I think, around this, of including sort of more patient perspectives in the evaluation of treatments. And so patient-reported outcomes you see referred to increasingly, although so far I think they've only been used in studies of one HIV-related treatment, which was the liver dystrophy treatment, a grifter. Another possibility might be one of the things that's being looked at now in the context of aging research, which is frailty, sort of weakness that can develop in old age and can be associated with illness and mortality. There's some indexes that try to sort of measure the amount of frailty, and it might be possible that an intervention could affect or improve that, but that's really kind of speculative because it hasn't really been looked at in this population of people with poor immune recovery. And then probably what seemed to be of most interest to the FDA was just whether there might be sort of clinical symptoms that are not quite as serious as the kinds of things that have generally been evaluated in studies, like serious non-AIDS events, cardiovascular disease, those kind of serious things, whether there might be less serious issues that could be evaluated and give some sense that an intervention was having a beneficial effect on health. One of the things that Matt Sharp talked about at the meeting, he gave some testimony about his participation in the Sangamo trial and sort of noting that he'd been having chronic issues with upper respiratory tract infections that really went away in association with the CD4 T cell recovery, which is anecdotal, but just might be a hint of the kinds of things that might be more frequent occurrences that could be evaluated in smaller trials to give an idea of whether the effect an intervention is having on the immune system is really going to be beneficial to somebody's health. And the FDA was pretty clear that although there was some attempt to maybe hope that there could be some surrogate markers of efficacy, they were very clear, I think, that based on the sort of IL-2 experience, that there would need to be some evidence of a health benefit for a therapeutic intervention to be appropriate for even a kind of provisional approval for people with poor immune recovery. And so then just the last thing is, what some potential next steps might be for us. I think it's been amazing to have so many people join the webinar, and I'm very grateful and thankful for people for joining. And one thing we could potentially do is have a follow-up webinar with scientists who have been involved in this work to give their perspective on what's going on. Perhaps we could consider, and if people would be willing, we could follow up by email to see if people might be willing to sign on to some kind of advocacy statement or article that articulates, the sort of global need, because this is a phenomenon that's seen around the world, and that makes an argument for trying to focus on this a little more, and particularly to develop interventions. I think that there's ample room to have continued dialogues with stakeholders, including the researchers, FDA, and industry. There's another gene therapy company called Calimmune that has sort of expressed some openness to thinking about this kind of research, although that like a lot of companies these days, that they're more focused on whether there might be potential for some kind of cure intervention with their approach, and we're certainly open to sort of other ideas that you might have.
Yeah, I wanted to add, Richard. We will be sending, obviously, the presentation to everybody that signed up. We'll be posting it in different groups on Facebook and everywhere else. And we will try to engage a few of you that may want to work on this issue. Obviously, there are very few of us focusing on this, and we need more help. I think one of the weaknesses that we have right now when it comes to data is that we have not good data on what is it like to live with lower CD4s after years of undetected viral load. Is this population having more, like Richard said, more upper respiratory issues, more diarrhea, more IBS, more pain-related issues, et cetera, et cetera? Nobody has really collected that data. So my dream is to spread a qualitative, obviously, scientific as we can do it, survey to collect data online from that population, because really there's no other better way to do so quickly, and hopefully have some validity of that data, since there's a lot of bias on surveys, and we can actually publish that data in any of the conferences, so that we bring this up. The fact that you don't have to go for a survival endpoint, but really even lower incidence of certain health issues that we may be living with low CD4 cells. So that's my closing statement. I think, Richard, you want to handle the questions? We have a bunch, and we have some hands that people have risen already. So I don't know how we're going to handle these many questions. So you go ahead. I'll give you the first try.
Sure thing, and I'll try my best. I'll quickly try and go through some of the chat comments, and then open the audio so the people with hands raised can ask their questions. I think just there's been some suggestions in the chat about the importance of focusing on the gut, and also potentially advocating legislatively for consideration of these issues, as well, just as not just with necessarily researchers and FDA, and the potential importance of collaborating with people who are working on HIV and aging, because there is sort of a lot of overlap in terms of what the risk factors might be and the potential consequences of persistently low CD4s. And there's also a question about whether stem cells can help renew the immune system, which they can do. Stem cell transplants are used in pretty serious illnesses to kind of repopulate the immune system. The challenge is whether they can be used in people that don't require stem cell transplants for things like cancer, because there can be a risk associated with them. And what Calimmune, the company I mentioned, looking at gene therapy, is doing research involving extraction and infusion and gene modification of stem cells. That's a trial that's happening in California. I think the results are due fairly soon. So hopefully we'll get a sense of that pretty quickly. And then there's a suggestion about maybe looking at existing cohorts for data on the less serious clinical consequences. And I know there have been people that have been emailing researchers to try and see if that data exists, without success so far. But I think it hasn't really been fully explored.
Yeah, and I think there's one more thing I wanted to comment on that. It takes funding and it takes manpower to go back retrospectively to look at data from cohorts. So what we need to advocate for is also for the NIH and other sources of grants to fund this kind of data mining from different cohorts that we've had in the past. The Kaiser, the VA system, Jeff Taylor proposed that as an excellent idea too. So, we just need contacts within that system where we can tap into some of, as I said, the data mining on cohorts of that. So as you can tell, just by reading the comments on the Q&A, there's so many possibilities for activity and activist ideas. So I'm very happy to see that. Richard, just take over from here. Thank you.
Sure. So I'm going to take a bit of a risk and unmute everybody, but just to try and keep things vaguely organized, I've got Noreen Griffin and David Miller with hands raised. If it's okay, I'll start with Noreen.
Yeah.
And
the conference has been unmuted.
Let's try to keep from speaking over each other.
Sure. If you want to ask your question, Noreen, go ahead.
From what I'm hearing from you, that you think at this point at least the FDA is going to be more receptive to small companies working with immune reconstitution issues than they have been in the past.
Well, they've always been receptive. I'm sorry, Richard, you go ahead if you want to.
Go ahead, Nelson. Go ahead.
Yeah, they've never not been receptive. I think nobody has really proposed the orphan drug indication for this population, and they were extremely receptive on that issue. So, to be honest with you,
there's really been no immune-based therapy.
Yeah.
And I guess that's where I'm asking if they're going to be more receptive.
The biologics area of the FDA is a little more challenging. What I have, to be honest with you, I've been a little bit down on, as you can tell, the meeting was in June of last year. We sent emails to industry, to different companies, with the meeting minutes, which obviously were very encouraging. And we did not really see a really good uptake, a very good response. So that I was very surprised about. I thought we would get more interest from industry. I think what really scares the industry is the endpoints, and Richard explained the different possibilities for endpoints. So I'm going to take Richard. Richard, just take over from this and add more to that.
Sure. No, I think that is more. Probably the FDA is not really. I mean, obviously they need the solid data to be sure that something works, but they're not really. They've not really been oppositional. It's really the companies and the industry that there hasn't been a massive amount of interest in trying to develop therapies for this population. I think that's been more where the issues lie, and just that, if it might be possible to do smaller studies with just CD4 counts or something, maybe there would be more interest. But I think the FDA's position, is reasonable, that if you can't be sure that a CD4 count is going to increase, is going to benefit somebody's health, then you don't really want to be approving something just based on that marker.
Okay. Thank you.
Sure. And David Miller?
Hi.
Go ahead.
So we're all aware of the data from the Journal of Virology that was published a few years ago on CD4 T cells, including Th17s, are intact in the gut mucosa of HIV-infected long-term non-progressors, the LTNPs. We know that this new emphasis on microbiome restoration is still in its infancy, despite the fact that there's a lot of promising data that was presented at IS 2017. Given that we know there's so much gut insult due to the antiretrovirals, despite how much progress has been made, and due to the fact that we're years away from a viable therapeutic for restoring the gut microbiome, why don't we start addressing the consideration that there's a large patient population that has differentiations in their ADME of the pharmacokinetics of antivirals due to unremittent diarrhea. We have therapies like Mytesi out there that can address that. We also have other probiotics like the one being done on BizBiome in the ACTGs, which might be helpful in combination to at least preserve the Th17 cell lines.
Sure. I think there is definitely, one thing that's happened over the last few years, I think, is that the whole area of the microbiome, the gut bacteria and its role in immunity and in drug metabolism has really gone from a somewhat sort of, it was a bit sidelined for a long time and people didn't really take it necessarily all that seriously, but I think now it's become far more mainstream, which is at least an important step in terms of getting the kinds of studies you're talking about done. And I think, there obviously meetings don't solve problems necessarily, but the fact that there's now an annual kind of research meeting about the microbiome, I find kind of encouraging, because at least it means that people are working to identify the key questions. And I think in this particular population of people with suboptimal immune recovery, that is still going to be a major focus, particularly around the use of probiotics.
What about the issue of the unremittent diarrhea and how that's contributing to gut inflammation? Because we know that's happening in a wide range of patient populations, and that's contributing not only to the malnutrition, but also the inflammation that's killing the Th17s, according to Shinosi's data.
Sure, and I think that is one of the outcomes that's been reported in some of the more encouraging studies, has been improved gut health and reduced diarrhea. I think there needs to be a more systematic evaluation of, because there's so many different kinds of probiotic. I think there needs to be an effort to really figure out which ones are the ones that are going to get you the most benefit. And that hasn't, so at least I think there is some discussion about that happening, but it hasn't really happened as yet, which is unfortunate. I think at the moment people are left trying to sort of guess what probiotic they might want to spend money on, which is not an ideal situation at all.
I get that, but just moving beyond the probiotic issue, because we're years away from that, not to belabor the point here, so I appreciate the dialogue. We have Mytesi on the market right now. It's hard as hell to get it prescribed, and a lot of clinicians aren't recognizing that intermittent or unremittent diarrhea is contributing to gut inflammation.
David, let me interject here. The goal of this meeting is not to talk about possible options for research. It's more about what you, as an activist, can do to help move this forward. So this is an advocacy and we're going to have another conference call with different researchers. The products you're mentioning are both FDA approved. They can be used. There may be some insurance-related reimbursement issues, but they can improve. So either the company has to spend a little bit of extra money on funding for immune reconstitution-related work, or get grants from NIH from different private investigators. So they're already available. And I would hate to talk about diarrhea throughout the questions and answers section of this call and let somebody else bring up other issues related to activist work. We need help. More than the different options that we should be testing, we need help on logistics and how to get researchers and even cohorts to provide some more data to build a case. Okay, so thank you very much. In the next question, Jeff Taylor, you want to say something? Are you on mute or not? He's probably on mute. Somebody else had their hands raised. It's not more longer there. Does anybody else have a question? Well, as I said, we'll be following up. Everybody provided their. Jeff is asking, how do I unmute? Richard, you're the admin. You're the admin.
I think you should be unmuted, Jeff.
Everybody, yeah. Everybody should be unmuted. Yeah. Right now, everybody on the phone or on the computer are unmuted. At least that's what the program is showing. He raised his hand. Anyways, we will be sending the slides, hopefully this recording, and some ideas on activist activities, and we really would appreciate them to get some feedback on how everybody can get involved in working with us on moving this forward.
One other question for Nelson and Jeff. What happened to the IP from the French company? Does anybody know? Is it gone?
The Cytheris IP it got. Our understanding is that it got transferred to the French ANRS, possibly in a collaboration with another company called. I forget the name. It was a small biotech. We've tried to find out from Yves Levy what the status is, but we have not been able to get a clear answer. Perhaps we can try and get him to be one of the researchers on the next call to help explain what's happening with that, because I think that is important to find out. And hopefully another potential target to try and encourage development of IL-7 is the Korean company that's developing the long-acting formulation. And just as the last thing, as people are signing off now, and we'll wrap up, in addition to the video of the webinar, we'll also post a version of the PDF of the slides that will have embedded links to the papers and the research that's referenced in the slides, so that there's a way to look further at the papers. And unless, Julie, is there anything else you want to add, Nelson?
No, I just want to thank everybody. A very great response. Spread the word.
The conference has been muted.
Oh.
The conference has been unmuted.
Oops, we're unmuted again. So thank you so much for your support, and we'll be following up on email, okay? And before you go, I'm going to plug Richard's Treatment Action Group's website, because there's a lot of good information on immune reconstitution that Richard has written there on his. There's a blog page that he writes summaries every week.
Just one more quick question before we all go. Is there any plans to put together a session at AIDS 2018? We're only 8 months away from Amsterdam. That's
a really good idea.
Are you proposing that you're going to get involved in that?
I'm proposing to raise the money for it, since IS doesn't do anything for free. You wanted to volunteer, I just stepped up.
There you go. Thank you. That's the kind of talk we want to hear.
And we will definitely.
I do want to talk about diarrhea in that one.
All right. Well, thank you very much, everybody.
Thanks, all.
Happy holidays.